morning, everyone. I'm delighted to be here. Thanks to the organizers for inviting me, and I'm excited to be able to tell you a little bit about some new work in our lab. This was just published this month in Genome Research, and it's the first time I'm talking about it. It's primarily the work of a senior graduate student who finished in November, named Jianling Zhang. So I probably don't need to tell this audience, but nucleosomes are abundant and fundamental across eukaryotic genomes. They're the most fundamental unit of packaging, and they play a role in regulating the accessibility of the genome to factors that are involved in replication and transcription. As you all know, but just to remind you, typically around 147 base pairs of DNA are wrapped around the histone octamer. There is an axis of symmetry called the dyad, which is the central position of the DNA. And if you rotate the nucleosome around that axis, then you essentially see the same kind of position. And everyone knows that identifying nucleosome positions is particularly important to understanding its role in these various regulatory processes. Now, historically, there have been two major uses of nucleases to profile chromatin. There are many others, but I'm going to focus here today on MNase and DNase. And by way of contrast, MNase is an endoexonuclease that's small enough to cut linker DNA between nucleosomes. So this has been the workhorse for positioning nucleosomes across genomes. You digest the genome with MNase. You isolate typically size-selected mononucleosomal bands and sequence them to position nucleosomes. This has been done extensively in yeast, but it's a little bit too expensive to do it in human. As far as we know, there's only one study that's attempted this, and the data is still quite noisy and hard to interpret. On the other hand, DNase has primarily been used to probe open chromatin. And it's been used extensively across the human genome. There are hundreds and hundreds of DNase experiments. Interestingly, there's only one DNase experiment in yeast before we did this experiment. This particular paper. There's this separation of roles where MNase is used to position nucleosomes and DNase is used to study the regions where nucleosomes aren't. And there's been a recent explosion in probing into the fine detail of these DNase hypersensitive sites and considering things like transcription factor footprinting. And my lab has done some work in that area, but I'm not going to tell you about transcription factors today. Today I'm going to tell you about a story that happens sometimes in science where you want to use data for one thing, and as you explore it a little bit, you discover something completely different, and you follow it to its natural conclusion, and something interesting happens. So just so you know, before we worked out the structure of the nucleosome with X-ray crystallography, people used to use DNase to probe its structure and very carefully study how it would nick the nucleosome. So there is some rich literature for understanding how DNase interacts with nucleosomal DNA, and I'll come back to that in a minute. So we asked the following question: could we use DNase to explore not just nucleosome-depleted regions of the genome, but actually nucleosome-associated regions of the genome? Now, why did we think this might possibly be feasible? First, there was a recent paper in 2012 by Brogard et al. Reporting very precise locations of nucleosomes. And they did this without using MNase. They used a different method in which they genetically modified histone H4 so that it would cleave the genome in very specific positions. And after a little bit of data processing, they were able to determine nucleosome positions down to single base pair resolution, which even with MNase, there's a little bit of a 2 to 3 base uncertainty, even if in the best of cases. So the precision was much higher. Also, collecting DNA-seq data of high depth and quality is fairly routine in these days. So we thought there was a lot of that available. And when we combined these, we thought maybe we could see something about how DNase interacted with the nucleosome because of these traditional papers from the 70s and 80s. And then maybe we could formulate a statistical model to position nucleosomes based on that signal. And what I'm going to describe today is our work in these directions. So here's the setup experimentally and the kind of data that we had available to us. As I mentioned, there was this chemical cleavage method that provided precise nucleosome positions. In particular, they called something called nucleosome center positions, and they had a score to say how confident they were about and how well positioned that nucleosome center was. And we used the top 2,000 of these. These were across the yeast genome. There were 67,000 nucleosomes reported, about 80% of the yeast genome, believed to be a fairly complete map. And we chose the top 2,000 to be our positive set, our training set, to do some exploratory analysis about how DNAse interacted with those nucleosomes. We ourselves, yes?
I just want a quick question for clarification.
Yes.
So these are done on populations of cells, right?
Correct.
And the nucleosomes may sit in different places.
Absolutely.
So when you talk about the position of a nucleosome,
Correct.
What do you mean with that? What do you have in mind?
What do we mean? So the cell population, each nucleosome will sample different locations. At different parts of the genome, those may be sampled with sort of increasing fuzziness. So in some parts of the genome, a nucleosome in different cells may hop all over the place, or in the same cell over time. You could think of it either way. And in other places, the nucleosome may be locked in a little bit more rigorously because of sequence reasons or because of barriers and things like that. So in their data, they were able to distinguish between a position of a nucleosome and the score is, roughly speaking, the amount of consistency across the population. So as that score goes up, you expect that nucleosome to be more strongly positioned, and that's why we focused on the top 2,000 instead of all nucleosomes, because some of them would be fuzzier and shifting around.
So it's basically the places in the genome where you're most likely to find this position covered by a nucleosome.
I guess the right way to think about it probably is that as this score goes up, you would expect a higher and higher fraction of the cells in the population to agree on the exact position of that nucleosome. But as the score fuzzes out a little bit, you see alternative positions, and the data, you'll see in the data that there are alternative positions, and they tend to be slightly shifted from one another. Okay. So we collected our own DNAse-seq data in yeast. This is in conjunction and collaboration with the Crawford lab. Greg is one of the authors on the paper as well. And just for comparison, to make sure that our results that we saw in our data were consistent, we also used the one data set that was available for yeast DNAse-seq, which was collected in the STAM lab. Jay Hesselberth was the first author. So what did we see when we looked at these 2,000 nucleosome center positions and summed up the DNAse cuts across those positions? We saw the following. Now, first, just as a small aside, everything that I'm going to show you is what we call transformed counts. This is a very simple transformation. We take the number of cuts mapped to a particular site, and we put it through the inverse hyperbolic sine function. Inverse hyperbolic sine, if you're not familiar with it, I commend it to you. It's essentially a log transform that behaves well at 0 and 1. Okay. So once we transform these counts, and this is mostly to do variance equalization and to get really tall, highly cut regions like the DHSs to be on a similar scale with the less cut regions of the genome, we saw this profile. And it has, or we observed, 3 basic components. There's some fine details. But first, you can see that the overall shape is quadratic, that you're more likely to cut away from the dyad than in the center of the dyad. And we believe this is largely because of DNA breathing and partial unwrapping of the nucleosome, such that there are times when the ends of the DNA are actually accessible for DNAse digestion because they've unwrapped from the nucleosome, making them more accessible in more places. Second, you see a strong oscillatory signal. And this also makes a lot of sense. This is consistent with this literature from the 70s and 80s. DNAse binds in the minor groove and occupies 8 to 10 bases in order to sit down. And as the DNA double helix wraps around the nucleosome, the minor groove becomes very exposed to the outer solution and then very buried. So back and forth the minor groove goes, and so the DNAse can cut here, but then it can't cut here, but then it can cut here. Okay? And one thing surprised us. Of course, you expect, because of the dyad and its axis of symmetry, for the orange curve read from right to left to agree with the green curve read from left to right. In fact, let me just show you that. If I flip the orange curve around, you see that they're in striking but not surprising agreement. But what was surprising to us was that as you move from the 5' to the 3' direction on either strand, or as you come into the dyad versus leave the dyad, there's an asymmetry of behavior. We have a couple of theories about this. The one that we think is most likely is just the fact that as you come around the first time, you're sort of on the top of the nucleosome and it's easier to bind. And as you come around the second time, there's steric occlusion from the first wrap of the DNA. And we think that that's the explanation. Regardless, it's a striking empirical pattern. And so we asked whether or not we could use it to position nucleosomes across the genome. However, we were a little bit concerned because nice work by Harman and Remo and also seen in our lab, we identified the fact that DNase does have a slight sequence bias. And we also know that DNA, as it wraps around nucleosomes, has a sequence pattern. And it has a periodicity of 10.3 bases in terms of these A's, AT dinucleotides and GC dinucleotides. And so we were concerned that possibly the signal we were seeing was just DNA's bias mapped to sequence patterns in the genome and reflected nothing of nucleosome positions. So we were able to reassure ourselves by looking at this data from the STAM lab. So this is the Hesselberth data. First, we reconstructed the same pattern that we saw in our data for nucleosomes, the same 2,000 positions in the yeast genome. But importantly, their data also included DNA's digestion of naked DNA without nucleosomes, and that's what these two curves are, the Watson and Crick strand patterns at those same locations when there's no nucleosome involvement. And you see that there's no strong oscillatory signal and that you see no quadratic shape. So our next question became, is the profile distinctive enough to tell, just by looking at DNA's data, whether there's a nucleosome there or not? And the first thing we did was consider this from a classification point of view. So we wanted to see if we could identify the 2,000 nucleosome positions from random locations in the genome and distinguish them in a classification manner. So let me tell you a little bit about the model and its notation. First, I'm just going to designate the cuts centered around a particular position in the genome on one strand by an X vector and on the other strand by a Y vector. All of our models are going to consider these two strands separately, but sharing the same model in the opposite direction, as you'll see. After the transformation I described, we observed that DNA's cuts tend to be normally distributed, and also that the variance is proportional to the mean. So we introduced this constant of proportionality and fit it. As it happens, it's not that interesting. It's pretty close to 1, and it's pretty consistent across the genome, so it's not a major thing. Then we wanted to discuss how the mean of each of these series would depend on or be related to the DNA's data, and we decided to parameterize that in the following way. We explicitly modeled the quadratic shape, so you have a level parameterized by A, a quadratic term parameterized by B, centered on the 0 point, and then Z reflects all the remainder, all the oscillatory terms that are superimposed upon the quadratic shape. And you'll see that we modeled the X series as being in the negative direction from the Y series, but X itself and Y itself are not symmetric about 0. When you're outside a nucleosome, we just assume there's some constant level of DNase cutting and no particular pattern. So that's a constant. That's a different A. OK, so with these two models, we can then write down the likelihood of the data for being within a nucleosome and the likelihood of the data for being without a nucleosome as two alternative hypotheses. This is nothing more than the product of the data at each position and then the data for each strand, and you see here a normal model. Similarly, outside a nucleosome, there's a normal model, each position and each strand. The difference here is just that here we have the constant model, and here we have the oscillatory profile. And
you assume that nucleosomes don't overlap in this model, or not?
we'll come back to that. In this particular case, we're going to assume that they don't. But let me make one point which might clarify the question a little bit. We're asking whether or not a position, a single nucleotide, represents the center of a nucleosome. Okay? So in the classification problem, when I said we picked 2,000 random locations, you might think, well, the genome's pretty covered in nucleosomes. And so the probability that you're going to find a spot that's covered by not covered by a nucleosome is pretty low. But it becomes clearer when you think the probability that a single nucleotide is the center of a nucleosome is pretty low. It's more like 1 in 200. So our negative set is not perfect, but to a first approximation, it's mostly non-centers of nucleosomes.
I was mostly thinking about sort of that you're looking at this population average. And some nucleosomes will not be well positioned, so
exactly.
The criteria of non-overlapping kind of
right.
doesn't really apply to the average.
I'll come back to that. So for now, we're going to just start a simple thing to see if we can distinguish nucleosomes from non-nucleosomes, and then later we'll use this to map across the genome, and we'll come back to whether or not they can overlap. That's a great question. Okay. Now, it turned out that so you could compare the ratio of those two things just to do a simple hypothesis test of which is more probable. It turned out that because the local cutting level of the DNAs across the genome is regionally variable, and there's some uncertainty about that, we considered a Bayes factor instead in which we integrate out this A parameter, so we don't fix it at its MLE estimate, but consider a distribution over possibilities. And as you'll see, we got slightly better results with that. I'll come back to that. So this is our final score. It's nothing but a description of the likelihood of the data with a nucleosome compared to the likelihood without a nucleosome, but in each case, integrating out the A parameter. And later on, when we come to the mapping question, all we're going to do is take this score and compute it for every position in the genome, and we'll get a curve, and then we'll break that curve down into positions of nucleosomes. So first, how does the classification work? Reasonably well, and we did it with both our data and Hasselberth's data. We felt pretty good after seeing this ROC curve that we were able to do a reasonably good job. Both the data agree when the nucleosomes are fairly easy to distinguish. As they become a little bit harder to identify, our data seem to be slightly better. I don't think it matters too much. Coming to the question of this Bayes factor versus the likelihood ratio, we tried a couple of different things. We started with the straight likelihood ratio, and we saw a lower AUC, just over 0.7. These last four columns, we considered what happened when you added the Bayes factor approach, and they're in two groups. First, in these two cases, we just fit the model with a quadratic. We ignored the oscillation, thinking maybe that was just too noisy. It turns out that you get better results when you add the oscillation in. There's some interesting data there that's important, and it helps you position and distinguish a nucleosome from a non-nucleosome. In addition, we also considered whether we should integrate out just A or A and B. Because A and B didn't perform markedly better than just integrating out A, and because it's computationally more expensive to do it, we just dropped that. So our final choice was this one. We use a Bayes factor, we add the oscillation, and we integrate out A.
How many more reads do you have compared to the oldest paper?
That is a good question. I don't know the number off the top of my head.
The question is, how much do you buy by?
Yeah, some of it could be from that. This is sometimes people put these up to say, hey, our method is way better than their data, or whatever. That's not really the point. It's more to say that the conclusions about which model to use are consistent across the data sets. We focused on our data. We confirmed that all the major things we saw were also present in the Hasselberth data, but we're not necessarily saying that one data set's better than another. And we had 83 million reads. I can't remember off the top of my head how many they had.
So when you don't integrate out B, how do you set it?
We set it as an MLE value in the training set.
Overall.
So we did a k-fold. We did a k-fold cross-validation, and so we set it on the training set and then tested on the test set. Yeah, the same thing with K. K is the ratio between the mean and the variance. We also just set that with an MLE value. Okay, so What's that?
We have 23 million.
23 million. So it's almost 4 times more data. So can we exploit this classifier to identify nucleosome positions? That became the next question. Not just classify, that's a pretty simple setting, but to actually map. So to set the stage, let me show you, for example, a arbitrary section of the yeast genome. You see 2 genes here. You see a promoter region in between. And this is the DNAse data raw before transform that you would typically see. As you can see, there's a burst of cutting here in the promoter of ESL1, and that's the open chromatin region that people usually focus their attention on. But what you can also see is that because of the depth, there's a few cuts all along the genome, and it doesn't seem like there's that much there. We applied our transformation so that we could put the 2 more on the same scale, and you see that it's still quite noisy. But maybe you start to think, oh, there's a little dip here and a corresponding dip here, and maybe there's something there. So it's not really obvious from the data, but the model is able to consider all of these positions and compute a score at every position, and that's what I'm showing here in green. This is our score computed at every position. And it was reassuring to us that it tended to peak at the same locations that were identified by Brogard and their method as the centers of nucleosomes. And this comes back to your question. These little centers can sometimes be separated by 10 bases. There's some uncertainty about where this specific nucleosome really is. And our score also has a 10 base pair wiggle that we've smoothed out here so you can see more clearly where the peaks are.
I still don't understand it. Why can't it be that I understand that there is a nucleosome that is sort of there
Yes.
in that position, but
Right.
it could be shifting across cells
Absolutely.
by a few base pairs left and right. So why use this notion that there is one position where the nucleosome...
We don't use that notion. We don't use that notion. We simply train on 2,000 positions that seem to be across the cell population enriched at a single location, not always at a single location. Maybe 80% of the cells have that nucleosome in that position. And that's simply to train our model. We don't insist that that's true later on. I'll come back to that. You'll see there's an important point here.
Yeah, it doesn't really matter for the result. It's just conceptually.
I am in complete agreement with you. In fact, what I'm not talking about today is some work in our lab that this is fitting into related to compete. And compete is a model that says factors are all over the place and they're dancing and they're picking different positions and they're moving. And it's a giant game of musical chairs and every cell is doing something different. So that it's entirely the paradigm that I hold. Great. So again, just to sanity check that this curve means something, we did the obvious thing of seeing what it looked like when you stacked it up at various locations, genome features like transcription start sites or origins of replication or transcription factors that are known to sort of fit in between nucleosomes. And you see that the composite pattern recapitulates things that have been reported in the literature many times, including, and this is just for comparison, this is what the Brogard centers stack up to. So you can see there's a pretty good agreement between all of these things. But that's just a curve. How do you sort of come to a prediction of the most likely positions of nucleosomes? So here we are. At this point, I'm going to come up with a map, which is at each position in the genome, I'm going to call the center of a nucleosome, which you could think of as the most likely position of a nucleosome in that general region. But one of the things, well, let me just show you what happens. So one of the things that happens here is when we draw these ovals, the way the algorithm works is it takes the highest peak across the entire genome, and it calls that a nucleosome and puts one. And once it puts one, it excludes that region from other nucleosomes, but one thing it does is it only excludes 117 bases, not 147. And the reason for that is because one nucleosome, even though you think the center is here, it could shift a few bases left or right, and as it shifts a few bases left or right, we don't want to knock out another perfectly legitimate nucleosome nearby. We also know that they partially unwrap So they don't always take up 147 bases. So, oh geez, okay. So you can see sometimes the predictions actually overlap. So what does this map look like in comparison to the workhorse for, yeah?
Sorry, if you go back to the last slide.
Yes.
So like the area around the promoter is an open region.
Correct.
We know that because of the
Yeah.
concept.
Yeah, there's a bit of a wider NFR, a little bit of a bigger space between these two and a gap here.
So would you call also the spaces between the other nucleosomes also open, even though you don't have enough cut sites in the...
Yeah, I mean, that's a question of semantics. I mean, you could say what they are is open to sufficient digestion by DNAse to be detected as gaps between nucleosomes. And they're open enough to MNAse for sure. That's why people use MNAse to cut them. DNAse has typically had a little more trouble in there, and that's why people get a lot more cuts in the open chromatin. But this is a question of categorizing a spectrum. Let's talk more afterwards because I'm behind. So if you compare the reported, you can think of it as the Viterbi parse or most likely positions of nucleosomes from the Brogard data with these various results that were reported in the last few years from the Pew lab, from the Segal and Whittam labs, from the Iyer lab, you see that MNAse does a pretty good job of mapping the reference map. And actually, Jiang and Pew combined all these data sets to produce a consensus map that does much better than any of them individually. And then when we compared our DNAse map to these maps, we saw that we were doing essentially as well. There are some small differences. We have lower false positive and false negative, but we're not sure how much of that is just from sequencing depth. But the point is, you can do just as well using DNAse as you can with MNAse. That's the point. But this is all in yeast. We were curious how this would scale up to other genomes and how much sequencing depth you would really need to see these little signals in the nucleosome-associated parts of the genome. So we subsampled our data, and we saw how the classifier would change its performance. This is our data now. We have about 7 reads per nucleotide, or per genome length. And then this is the performance dropping off. And as a rough rule of thumb, we thought it was reasonable to think you need about as many reads as the size of your genome to do reasonably well. So that's the ballpark. That's hard to accomplish in human. It's trivial in yeast. It's pretty easy in worm and fly. But it's pretty hard in mouse and and human. But people have collected so many DNAse data sets that we pooled data. So there were 70 cell lines of the same cell from 70 different cell type from 70 different individuals. We pooled them together. We applied our model from yeast, so no fitting to human. And this is the curve that we get. And if we map nucleosome positions, these are the ones that we get. This is the one MNAse paper that's been done in human. And you can see that you can also learn positions. And the agreement is not quite as good. It's about 2/3 overlap. 1/3 we have a nucleosome where they don't. 1/3 they have a nucleosome where we don't. But in both cases, our data is limited. It's less than 1 read per nucleotide. Their data is also quite noisy. So there's still room to go in the human case. Coming back to the yeast data, there was one more thing we observed when we looked at how far off our map was from the reference map. And when we computed the center-to-center distance between the reference map called nucleosomes and our called nucleosomes, we saw a very interesting thing. With all of the MNAse maps, either individually or collectively, you see that if you position a nucleosome wrong, you shift it by a little bit, and then you're less likely to shift it by a little bit more. So you get a unimodal distribution that falls off smoothly. But when you position a nucleosome differently in a DNAse map versus the reference map, you're almost always going to be off by a multiple of 10. And this makes sense because there's this oscillatory signal, and if you don't get it right here, then the best match might be 1 shift over. And so what we concluded was even if we're not getting the center right, and maybe there is no the center, that there are alternative locations and that those are 10 base pairs apart. And so we can assess for the whole genome whether or not this point in the genome, when it's associated with a nucleosome, is likely to have its minor groove exposed or its major groove exposed. Because if a nucleosome is there, it will be in these sort of discrete positions, more likely than not. And so I'll say this quickly because I'm out of time. But with that in mind, you can essentially map across the entire genome this oscillatory pattern and ask whether or not this nucleotide is likely to be exposed as a minor groove or exposed as a major groove if it happens to be associated with a nucleosome. And we looked at transcription factor binding sites and that were within nucleosomes, and we found that they were more consistently aligned with this pattern than you would expect by chance. So the black curve is the oscillation, minor groove to major groove, of the motif's center, and the blue are random curves of the same size set and the same properties, but random. And I'll just close with this, which is to say that we looked at a number of transcription factors in yeast, which are in blue, or human, which are in red, and we empirically observed that the center of their binding site tends to be quite aligned with either the major groove being exposed by the nucleosome or the minor groove being exposed by the nucleosome. More than you might expect. It's sort of barren in the middle. And one interesting point is that if a factor is a minor groove binder, it's not necessarily the case that its binding sites are minor groove exposed by the nucleosome. Sometimes they agree and sometimes they don't. And at first that puzzled us, and then we thought, well, this may be part of regulation, right? These binding sites may fit into the genome in ways that sometimes the nucleosomes obscure them from binding, and sometimes the nucleosomes present them to be bound. And maybe, this is all speculation now, no data to show this, but maybe there's a connection between what we call pioneer factors, which can bind in nucleosome-associated DNA more easily, and a phenomenon related to this, but we haven't explored it too deeply. Let me just say thank you to my co-authors, to the great environment at Duke for doing this kind of work, for funding through the years that's made this all possible. And as I take questions, all of our software is available online in GitHub, and we have pre-computed tracks of this nucleosome score across the yeast and human genomes that you can download and explore if you want. And with that, I'll take questions.
Does your data predict any relatively long stretches without nucleosomes? I mean, would you see that in your model?
So the answer to the question is, do our data show long stretches of non-nucleosomal DNA? The answer is that it would be hard to tell at this point, and here's the reason why. In yeast, we have sufficient depth to cover the whole genome and make calls. Like we predict 67,000 nucleosomes and cover the genome just like you would expect a good nucleosome map to do. Some of you may be familiar, for example, with the newly published Nucleoattack paper that does something similar, try to predict nucleosomes from ATAC-seq data. It's a nice paper. It's a great method. But they only predict about 13,000 nucleosomes, so they're not covering the yeast genome. However, the yeast genome is known to be pretty well packed, and maybe the question isn't quite as relevant in the yeast genome. In the human genome, where it might be an interesting question, our data is of such low depth that there are swaths of the genome with almost no reads, but we don't predict nucleosomes there, but it's probably more a limitation of the data. So to really answer your question, I think depending on the organism you're interested in, for that organism, you'd want probably 4 or 5 reads per nucleotide, and then you could ask that question. So we can't answer it yet.
So essentially, the method can't really deal with regions that are not DHS accessible. Is that what you were just saying? So that some regions...
No, I think the way to think of it is that this is not, unlike so DNAse flash and Nucleoattack, those are methods that can position nucleosomes also, but they do it near DHSs. Ours is genome-wide, but you need enough depth that you actually are seeing reads outside of the DHSs.
That's exactly what I mean. So that you need to sequence much deeper, essentially. Because
yes. If you sequence at a depth that the only reads you see are in DHSs, you're not going to be able to conclude anything in between them. If you sequence at sufficient depth that the majority of your reads are in DHSs, but you also see a smattering of reads everywhere else, we're good. Bill.
When you tune the assay, presumably
yes.
that's partly because
yes.
the data was designed to try to find DHSs. So
exactly.
you tune the assay to do it differently.
Yeah, there's ways you could do selection of either based on length or isolation of various things. Even something like the digestion protocol could be tweaked. There could be capture technologies like exome sequencing that might be able to guide the pool towards parts of the genome. Yeah, that's exactly right.
Let's take
And we haven't done any of that tuning.
during the break, and let's
Great.
thank Alex once again.